On-Chip Droplet Culture for studying stem cell differentiation? Can miniaturized droplets on a chip replicate the complex environment needed to guide stem cells into becoming specialized tissues? Stem cells are nature's raw material, capable of transforming into any cell type in the body, from neurons to muscle fibers to blood vessels. Their ability to differentiate makes them a central focus in regenerative medicine, developmental biology, and disease modeling. But controlling stem cell fate, guiding them down specific paths to become exactly the type of cell needed, is a major scientific challenge. Traditionally, researchers have used Petri dishes or multi-well plates, relying on bulk cultures and chemical gradients to study how different environments influence cell fate. But now a new approach is taking shape: on-chip droplet culture. This technique combines microfluidics and droplet-based compartmentalization, allowing scientists to study stem cell behavior in a precisely controlled, miniaturized environment. But how can something as tiny and simple as a droplet, just nanoliters in volume, serve as a platform to unravel one of biology's most intricate processes? The secret lies in control and isolation. In a droplet microfluidic chip, each droplet acts as an independent bioreactor. By encapsulating individual stem cells or small colonies within droplets, researchers can tightly regulate the chemical and physical cues those cells experience. They can tune the concentration of growth factors, introduce morphogens with gradients, manipulate oxygen levels, or even add mechanical forces, all within a highly confined and reproducible system. Why is this valuable? Because stem cell differentiation is exquisitely sensitive to microenvironmental signals, often referred to as the cell's niche. In a standard culture dish, those signals are averaged out and subtle differences between cells can be lost. But in droplet microenvironments, heterogeneity becomes measurable. Scientists can monitor how one droplet containing one set of conditions leads to differentiation into neurons, while another leads to cardiac cells, all in parallel, all on the same chip. Moreover, on-chip droplet systems enable high-throughput screening. Thousands of different differentiation conditions can be tested simultaneously, each droplet representing a unique combination of time, concentration, and signal. This drastically accelerates the discovery of protocols for producing specific cell types, such as insulin-producing B cells for diabetes or dopaminergic neurons for Parkinson's therapy. You might wonder, don't such tiny volumes limit nutrient availability or waste removal? That's a good question. Researchers have addressed this by developing droplet incubation systems that allow for continuous media exchange, co-encapsulation with support cells, or dynamic droplet merging, mimicking biological systems where cells interact with fluctuating surroundings. Some chips even allow the droplets to be fused or split, letting researchers change the conditions mid-experiment to simulate the dynamic nature of development. Another fascinating advantage is single-cell resolution. By placing just one stem cell in a droplet, scientists can watch in real time as it divides, differentiates, or responds to stimuli. This is crucial for understanding how decisions are made at the cellular level, how identical-looking cells begin to choose different fates based on slight environmental nudges or stochastic internal changes. Beyond basic science, on-chip droplet culture systems are being adapted for patient stem cells such as induced pluripotent stem cells, iPSCs. This opens the door to personalized medicine where one chip can test how a patient's own stem cells respond to drugs, grow into tissue, or repair damage before any treatment is given in vivo. So what does this innovation tell us? That big questions in biology don't always require big systems. Sometimes the answers lie in tiny droplets, engineered with precision and imagination. On-chip droplet culture offers a scalable, sensitive, and remarkably versatile way to study how cells become tissues, how the body builds itself from scratch, and how we might someday do the same from a chip, one droplet at a time. The on-chip droplet culture paradox highlights how tiny, isolated liquid droplets, on the scale of nanoliters to picoliters, can serve as powerful microenvironments for stem cell growth and differentiation, mimicking complex tissue niches despite their miniature size. It challenges the traditional view that large, bulk cultures are needed to control stem cell fate and simulate biological complexity. Instead, this microfluidic approach reveals that highly confined droplets, when designed carefully, can offer more precise, reproducible, and efficient models of stem cell behavior, all while dramatically reducing reagent use and enabling real-time observation. Researchers have cultured human embryonic stem cells in microdroplets on a lab-on-chip device, monitoring how different signaling molecules influence their fate in isolated environments. It's like turning an Olympic swimming pool into thousands of tiny aquariums, each hosting a different ecosystem. More manageable, yet more complex and revealing. Droplet-based microfluidics refers to the generation and manipulation of discrete aqueous droplets within an immiscible carrier fluid, typically oil, using microfabricated channels. These droplets act as miniature reaction vessels, each with defined composition, temperature, and flow conditions. For stem cell research, this enables the creation of individualized microenvironments, or synthetic niches, that can mimic specific combinations of growth factors, extracellular matrix, and mechanical cues. Differentiation of mesenchymal stem cells has been studied by varying the droplet contents, some with bone-inducing factors, others with adipogenic signals, all in parallel on a single chip. It's like sending identical twins into separate rooms with different music, lighting, and food, then seeing which one becomes a dancer or a chef. Within droplets, diffusion, surface tension, and spatial confinement become powerful tools to influence cellular behavior. Cells experience localized gradients, high surface-to-volume ratios, and precise nutrient delivery, all of which affect fate decisions. By dynamically adjusting the droplet composition, flow rate, or co-encapsulation of signaling molecules, researchers can trigger specific differentiation pathways while minimizing cross-contamination or signal dilution. By embedding stem cells in droplets containing controlled ratios of Wnt, BMP, or Notch pathway modulators, researchers can drive them toward neural, cardiac, or epithelial lineages with high efficiency. It's like seasoning identical dishes with different spices. Each droplet becomes a culinary test kitchen, and the cell's flavor evolves with the recipe. On-Chip Droplet Culture reveals how complex biological behavior can emerge from minimal modular systems. It supports the study of emergent cell fate decisions, stochasticity in gene expression, and single-cell responses to microenvironmental changes. This platform bridges the gap between reductionist molecular biology and systems-level tissue engineering by enabling high-throughput combinatorial exploration of conditions influencing development and differentiation. Stem cells cultured in thousands of unique droplets on a chip can be barcoded and tracked to reveal how subtle differences in mechanical stiffness or ligand exposure lead to divergent developmental paths. It's like rolling 1,000 identical dice in different colored bowls and discovering that the bowl's color subtly shifts how the dice land. The droplets also act as metabolic micro-reactors, where stem cell energy usage, oxygen gradients, and metabolite concentrations can be closely monitored or tuned. Because the droplet volume is small, metabolic changes occur rapidly and can be detected with high temporal resolution. This allows researchers to correlate metabolic shifts with lineage decisions, uncovering how energy flow, mitochondrial activity, and redox states drive stem cell fate. Researchers have used fluorescent probes inside droplets to monitor lactate production and mitochondrial potential during the early stages of cardiomyocyte differentiation. It's like putting a tiny engine in a sealed dome. You can watch the smoke, heat, and fume build up in real time, showing exactly how the machine behaves under load. Each droplet acts as an information-rich micro-experiment, enabling high-constant screening of stem cell fate landscapes. By varying just 1 or 2 parameters across thousands of droplets, researchers can build multi-dimensional maps of differentiation outcomes, detect rare cell states, and test hypotheses with statistical rigor. This dramatically increases the data density per unit of biological material, transforming stem cell biology into a more computable, design-driven field. In droplet-based lineage tracing, fluorescent barcodes allow scientists to follow how individual stem cells progress through multiple fate decisions under tightly controlled conditions. It's like using a telescope on every grain of sand in a desert. Each droplet offers a world of data that once required entire culture dishes. On-chip droplet systems open new frontiers in personalized regenerative medicine, where patient-specific stem cells can be screened for differentiation potential, disease modeling, or drug response. They also enable the rapid prototyping of organoid models, allowing researchers to test different tissue engineering strategies or identify key signaling checkpoints in organ development. In pharmaceutical research, this method enables high-throughput screening of stem cell responses to therapeutic compounds, reducing cost and improving reproducibility. A single microfluidic chip can test how patient-degrade-induced pluripotent stem cells respond to hundreds of drug combinations across droplets, accelerating discovery for neurodegenerative diseases. It's like simulating 1,000 personalized treatments in parallel and watching which ones guide cells to heal, grow, or renew. Despite its power, droplet culture has limitations. Maintaining long-term cell viability, dynamic control of droplet environments, and integrating readouts at high resolution across thousands of units remain technically demanding. Merging droplet systems with optical imaging, RNA sequencing, and AI-driven pattern recognition is essential for moving from discovery to application. Engineering the chip architecture to maintain oxygenation, waste removal, and controlled mechanical stimulation is an ongoing frontier. Researchers are developing hybrid chips with embedded sensors, gel encapsulation, or fluidic multiplexers to allow extended culture and real-time feedback. It's like building a smart terrarium that monitors every plant's growth, water needs, and sunshine, all within a glass bead. In the future, on-chip droplet culture will evolve toward interactive, AI-controlled microsystems that autonomously adjust conditions in response to cell behavior. We may see self-learning chips that evolve tissue models, respond to external signals, or even test hypotheses on their own. The convergence of droplet microfluidics, synthetic biology, and machine learning will give rise to living labs on a chip, enabling predictive design of tissues, organs, and cellular therapies. The fundamental insight is that complexity in biology can emerge not from scale but from precision, modularity, and informational control. Startups and academic labs are now developing microfluidic chips capable of testing patient stem cell responses in real time, helping guide custom regenerative treatments or predict developmental outcomes in embryo. It's like turning the Petri dish into a quantum computer of biology. Each droplet becomes a living bit of intelligence capable of learning, sensing, and evolving.